Our project was a lab behavioral ecology experiment focusing on mate attraction in painted lady butterflies dependent on color preference in relation to mating success.
According to a peer-reviewed study by Daryl J. Kemp, several behavioral studies, such as those conducted by Stride in 1958, Silberglide and Taylor in 1978, Ferdisi and others in 2002, Ehlers and Boog in 2003, and Robertson and Montero in 2005, have provided evidence for color-based mate choice in butterflies. These investigations suggest that the pronounced coloration observed in male and occasionally females is a result of mate preference. Notably, male butterflies are often more conspicuous than conspecific females. Dating back to the days of Darwin in 1874, many researchers have speculated the vivid wing coloration is a consequence of female mating preference for exaggerated visual signals. Some alternative perspectives exist, as seen in Silberglide in 1984. This idea gains wide support from the observation and evidence that bright colors are often confined to the dorsal wing surfaces of males. Based on scientific articles read in regard to our research question, we developed the hypothesis that Vanessa cardui, also known as the painted lady butterfly, with brighter and more intense patterns on their wings, have greater reproductive success than those with less intense coloring. Our prediction states that Vanessa cardui will display a heightened affinity for the model butterfly of the species, showcasing more vibrant and intricate wing color and pattern. Due to this, it is anticipated that the individual test subjects will spend a greater amount of time within the designated test area where those replicated visual stimuli are present.
Our painted lady butterfly larvae were shipped in from Carolina.com, which, along with their food and butterfly nets, were shipped to Bridgewater College. Once the larvae arrived on campus, they were stored at 71 to 72 degrees Fahrenheit in McKinney Room 124 beside a timed light source in order to grow as needed. Their food that came with them in the shipment was given once a day until each butterfly made up their cocoon. Once they emerged as adults, sugar water was waiting for them in 2 petri dishes at the bottom of their butterfly net to be eaten as needed. The sugar water was also changed once a day, once the adults were started to emerge. For data collection, 2 different butterfly models were put on each side of a tank, one model with a dull wing pattern and on the other side a model with a brighter wing pattern. Each butterfly was assimilated in the middle of the tank for 2 minutes, and then the blinds were removed and we studied the subject for 3 minutes each. Each model was switched after each trial. The tank was also sanitized to prevent any issues with results or scents left behind to deter the choice in mating. Our pilot study allowed us to tweak a couple of things that we needed in order to make the study work better for the next test subjects. The large tank was set up, divided into 3 sections: the middle, the side closest to the brighter butterfly model, and the side closest to the duller colored butterfly. Every 10 seconds, a tick mark was placed in the section that the butterfly was in that time of collection over the course of 3 minutes. We had a total of 21 butterflies. Our graph shows the painted lady butterfly time near their preferred mate in the clear tank. On the x-axis, you can see the average time that each butterfly spent in the part of the tank over the course of 3 minutes. On our y-axis, you can see the number of stays that a particular butterfly had in each side of the tank. The graph shows the time that each of our butterflies spent each designated spot of the tank. Our sample size for the study was 21 butterflies. Standard error bars are shown and suggest that our mean values are less variable from the mean, suggesting all of our values are close to support our hypothesis that these butterflies prefer the brighter wing color over the duller wing pattern. Many did spend a lot of time in the middle of the tank, and for future researchers we would suggest that maybe longer assimilation time for the butterflies would be beneficial, or maybe a larger amount of models on each side of the tank would be beneficial in getting more results.
There is much significance in regard to this research. While numerous studies have been inspired by the brightness and chromaticity of butterfly wing coloration, comparatively little is understood about the selective factors driving the most extreme visual exaggeration in this group, when compared to other vibrant animal groups such as birds, fishes, and amphibians. These butterflies are useful in studying mating preference in regard to visual cues. From a study by Kemp in 2007, color-based mate choice studies in butterflies suggest that exaggerated male coloration results from mate preference from the female, which tells us that females aren't always the ones that are choosy when it comes to mate preference. According to Robertson and others from a 2005 study, sexual and natural selection pressures both are thought to contribute to characteristics and wing patterns of butterflies. This species made an excellent subject due to their intricate wing patterns and easily sustained diet. Our research showed that there was in fact a correlation between wing coloration and attraction among painted lady butterfly.